In the ocean's twilight, a silver hatchetfish turns sideways and seems to vanish from the water. Its body has not become transparent, and the surrounding darkness is not complete. Instead, flattened flanks, reflective surfaces, and downward light organs manage different viewing angles at once. The disappearing act belongs to real midwater fishes within the family Sternoptychidae, including species of Argyropelecus. Their deep blade-shaped bodies resemble small hatchets, giving the group its common name. Predators may search from above, below, or beside them across a habitat without rocks or vegetation. Camouflage must therefore work against faint skylight overhead and diffuse blue illumination around the body. Direct measurements establish reflective anatomy and bioluminescent photophores, while effectiveness varies with posture and conditions. History is how one small fish reduces several kinds of visual contrast without possessing a single invisibility cloak. From the side, mirror-like skin can return the color and brightness of neighboring water. From below, controlled ventral light can soften the dark silhouette that its body would otherwise cast. If you watched from one fixed angle, disappearance could look perfect, while another observer still detected the fish. The investigation follows changing light downward, then tests each camouflage layer against a predator's possible view. No legend is required, because optical measurements and anatomy already reveal a sophisticated compromise. Hatchetfish disappear only approximately through matched signals that reduce detection rather than abolish it. Sunlight entering seawater is absorbed and scattered, with red wavelengths disappearing faster than blue. By twilight zone depths, the remaining downwelling field is dim, predominantly blue and strongly directional. Brightness overhead exceeds brightness below, producing silhouettes when animals are viewed against the residual surface glow. From the side, surrounding light is weaker and more diffuse, changing both color and polarization. A uniform pigment cannot match every direction across this uneven visual field. Marine hatchetfishes solve the problem partly through extremely compressed bodies, with narrow frontal and rear profiles. Reduced outline means less area interrupts downwelling light when the fish maintains favorable orientation. Their flanks contain layered, reflective structures that return incident light rather than absorbing it as a dark patch. Guanine crystals contribute to this silvery reflectance, a widespread optical material among fishes. The arrangement of reflecting layers influences which wavelengths and angles are returned. Biological reflectors can spread reflection across angles and wavelengths, reducing conspicuous peaks under natural illumination. Specific optical performance differs among tissues and species, so one laboratory measurement cannot describe every hatchetfish. Still, the mainstream interpretation is robust. Lateral reflectance helps match the surrounding light field. The fish's blade-shaped profile and reflective sides function together rather than as independent decorative traits. When it rolls, the match can fail, creating a flash that reveals movement. Maintaining posture therefore becomes part of camouflage, linking muscle control with optical anatomy. Small fin adjustments stabilize the body while minimizing sudden accelerations that would expose changing surfaces. If you imagine a mirror drifting through a blue fog, angle matters as much as color. The hatchetfish must keep reflected radiance close to the background, reaching a viewer from the same direction. A predator perceives contrast, and reducing contrast shortens the distance at which an outline can be detected. That benefit is probabilistic because eyes, water clarity, and viewing geometry constantly change. Side reflectance addresses only one part of the animal's three-dimensional visibility problem. Viewed from below, even a narrow body blocks downwelling light and forms a darker patch. Hatchetfishes carry rows of photophores along their ventral surfaces that produce blue-green bioluminescence. This counterillumination adds light where a silhouette would otherwise appear against the brighter water above. Experiments on related midwater fishes demonstrate control over emitted intensity in response to ambient illumination. For hatchetfishes, anatomical and physiological evidence supports adjustable counterillumination, though natural performance is difficult to film. The photophores point primarily downward and include optical structures that shape their output. Their spectrum broadly resembles the wavelengths traveling farthest through clear ocean water. A close match in color without a brightness match would still leave either a dark or luminous target. Intensity regulation is therefore central to the hypothesis that these organs conceal rather than merely signal. Ambient light changes with depth, cloud cover, time, water properties, and the shadow of nearby organisms. The fish must update its output as conditions shift, probably through sensory and neural control. Exact response times and control rules vary and remain incompletely resolved across the family. When you follow a hatchetfish descending, its camouflage becomes a continuously adjusted calculation. Downwelling light weakens, so excessive ventral glow would transform concealment into a beacon. Photophore output must fall accordingly, while lateral reflectors continue matching dimmer neighboring water. At greater depths, bioluminescent flashes from other organisms may become more important than residual sunlight. Counterillumination is most useful where an overhead light field remains strong enough to create silhouettes. It cannot erase the fish when viewed against an unrelated flash or illuminated directly at close range. Camouflage reduces one sensory route and therefore changes odds rather than guaranteeing survival. Another limitation arises when the fish feeds, turns, or flees and can no longer maintain ideal orientation. Hatchetfishes eat small crustaceans and other midwater prey, requiring movements that temporarily compromise optical alignment. A detected prey item provides a cue, and the fish must leave stable posture to intercept it. Rapid acceleration may generate a visible flash, but hesitation can allow the meal to escape. Success supplies energy. Failure spends energy and may expose the hunter to its own predators. This cue-action-consequence sequence makes camouflage and feeding competing demands within the same second. Predator approach creates a different decision, sometimes favoring immobility before an explosive escape. Holding orientation preserves the optical match until movement becomes less risky than remaining nearby. The timing of that transition is difficult to observe naturally and should not be stated as a fixed rule. Hatchetfish camouflage operates within the twilight zone's great daily migration. Many individuals move upward at night toward richer feeding layers and descend as daylight returns. As you trace that route, ambient brightness changes by orders of magnitude and viewing backgrounds continually rearrange. Reflective flanks need no metabolic light production, but counterillumination consumes biochemical resources and demands regulation. Together, passive and active systems spread the cost across different viewing directions. Reduced detection helps hatchetfishes approach prey while also avoiding larger fishes, squids, and marine mammals. This dual benefit can influence when they migrate, how long they feed, and which depths remain tolerable. By consuming zooplankton and becoming prey, they transfer carbon through midwater food webs. Their vertical movements also transport material downward through respiration, waste, and mortality. Camouflage thus affects more than individual survival. It shapes encounters within a major planetary migration. The strategy may weaken as artificial light from vehicles suddenly overwhelms the natural field. Animals filmed under submersible lamps can appear bright, dark, or flashing in ways they would not naturally. Collected specimens lose posture, tissue hydration, and living control of photophores. Photographs at the surface, therefore, demonstrate anatomy, but cannot alone demonstrate underwater invisibility. Researchers combine optical measurements, anatomical examination, behavioral recordings, and environmental light profiles. They also reveal variation, because different hatchetfish species occupy different depths and possess distinct reflector arrangements. This optical diversity allows related fishes to occupy distinct portions of a changing midwater light field. Their survival affects zooplankton consumption, predator diets, and carbon transport through the daily vertical migration. For human observers, the camouflage demonstrates why vehicle lighting can reveal an animal while concealing its natural appearance. The ecological achievement is measured in delayed detection, not in a perfect disappearance staged for a camera. Behavioral recordings could show how feeding and escape interrupt the best optical orientation. Repeated trials under natural light would distinguish typical performance from one unusually favorable disappearance. They could identify the distances where camouflage meaningfully delays detection rather than merely impressing human observers. Comparisons among Argyropelechus species could connect habitat depth with reflector structure and photophore control. Researchers must still avoid equating reduced camera contrast with invisibility to every animal eye. If you return to the opening twilight, the silver blade is still present within the blue water. Its narrow edge minimizes obstruction, while its flanks return fragments of the surrounding field. Below, photophores replace a measured portion of light its body has blocked. The match shifts as the fish turns, descends, feeds, and encounters passing shadows. Direct evidence establishes these structures and their capacity to manipulate reflected and emitted light. Mainstream interpretation identifies lateral matching and counterillumination as complementary camouflage. The precise survival advantage under every natural condition remains harder to quantify. Hatchetfishes do not literally disappear. They reduce the visual differences by which a watcher could find them. Their concealment is strongest when anatomy, posture, emission, and ocean light briefly align. In that dim alignment, the opening silverfish becomes not invisible, but scientifically difficult to distinguish from the sea.